All right, we're going to go through some stuff that we talked about last week and some new stuff. Eventually, I want to get to DNA technologies. So, I'm going to talk more about how DNA works and how it can be changed and controlled. So, last time I talked about DNA structure and how DNA is replicated. And the two strands are separated by enzymes, and then a single enzyme, DNA polymerase, adds complementary bases to one of the old separated strands, which serves as a template to produce two new strands that are identical. In bacteria, this is the simplest way that this happens is by separating these strands at the origin of replication and rolling off a circle to produce two new strands that happen when a DNA is, or when a bacterial cell is about to divide. Then the other main thing we talked about was the process of taking DNA, the information in DNA, and producing the chemicals that lead to physical traits. This is the central dogma where DNA is copied during transcription into a messenger RNA, and then that initiates the process of translation where a protein is made based on the DNA code. Transcription is a lot like DNA replication. A portion, though, only a portion of the DNA molecule is separated, and so there's this transcription bubble which begins at a particular site, and then an enzyme, RNA polymerase, starts building a complementary strand to the template DNA strand. But its complementary strand, instead of being DNA, is made up of RNA nucleotides. Here, uracil replaces thymine in forming a pair bond with adenine. And the RNA polymerase is just, adding complementary nucleotides. Then the messenger RNA in translation is going to use ribosomes and transfer RNAs to assemble a protein. Okay, the transfer RNA reads the anticodons, the three base units on the messenger RNA, and it takes those three base codons and it converts them to amino acids. And so there are 64 codons, and some of them code for multiple codons code for the same amino acid. But there are some special codons, the start codon, UAG, and then the stop codons, and there are three of those. And so the start codon codes for methionine, MET shown here, and so okay, all right. Okay, so let's go to the next slide. So here shows translation in action, where the ribosome has binded to the messenger RNA, and as it moves along the messenger RNA, transfer RNAs recognize anticodons, or recognize their anticodons, recognize the codons on the messenger RNA, and bring the appropriate amino acids, which the ribosomes link together. Eventually, they'll get to the end of the messenger RNA, and there'll be a stop signal there, and the ribosome will release the new protein, and the messenger RNA can then be reused. And so this is how traits are produced. The DNA information is made into a messenger RNA, and then the messenger RNA is made into a protein using the mechanism of the cell. And I should point out that these inherited genotypes that we get and that cells get from their predecessors, it's not the end of the story. That different environmental conditions can produce different phenotypes. And this happens because of the control of protein synthesis. The gene is the structural gene, and that's the part that's made into the protein. But there are also regulatory parts of the gene. These include sequences that are just upstream or ahead. I've talked a little bit about the promoter. That's the part of the gene, or the regulatory part of the gene, that the RNA polymerase recognizes. But then there's also an operator portion here shown in the lower one. you can see the operator can bind to repressors that block the RNA polymerase from moving forward. And so this repressor, this can lead to repression where the gene is not turned on unless it's needed. And repressors sometimes will bind to a specific molecule that will then allow the repressor to release the gene, and that allows the RNA polymerase to then go on and make the messenger RNA and make the protein. We have so both repression and induction. We can control what genes are active at what times. And this allows cells to only make the proteins when they need it. And so this is a form of negative feedback control that allows cells to control which proteins are made. Okay, in addition, I talked about mutations can change the structure of the gene, maybe just a little, or sometimes if they make a stop codon, can change it a lot. And so genes can change over generations through mutation. And cells can also acquire new genes through horizontal gene transfer, mechanisms that allow cells to get whole new genes transferred from other organisms. The first of these mechanisms is transformation, where cells take up DNA from their environment and incorporate it in their chromosome. They only do this at some times, and not all cells will do this at the same time, but this allows cells that are under stress to get new DNA and to try out new traits. And this can even happen between cells of different species. Often these are genes like for pathogenicity or sometimes antibiotic resistance. In other cases, a whole series of genes can be transferred in conjugation, where genes allow the cells to make a bridge between a cell having the gene and a new cell, and then a whole series of genes can be transferred. Again, occurring sometimes, usually between the members of the same species, but sometimes between members of other species. And then in transduction, we get the movement of genes due to a bacteriophage that incorporates a bit of host chromosome accidentally. And again, this doesn't happen every time, but bacteria have very, very high populations, and there's large numbers of phages, and so this can happen at a surprisingly common rate. Okay, once scientists understood all these things, they began to be able to understand that these processes were controlled by certain chemicals and certain enzymes. And they began to develop DNA technologies using these things. And this is just manipulating cells and DNA by using a knowledge of how cells work. Usually this requires that scientists have isolated enzymes that manipulate the DNA and gotten them to work in test tubes. Often these processes are then automated, and using computer analysis can be carried out in large scale. DNA technologies include genetic engineering, DNA sequencing, something called the polymerase chain reaction, using DNA or molecular probes to find molecules, and many, many other techniques. This woman here, she's doing DNA sequencing. Okay, and it's an automated process using an enzyme that we've isolated from cells, DNA polymerase. And so we take in DNA that we want sequenced, we put in some primers and the enzyme, and then we add nucleotides, the regular nucleotides and some modified nucleotides that have a radioactive tag or a probe that we can find. And these are our chain terminators. And so this DNA polymerase will work until it uses one of these chain terminators, and then it'll stop replicating and release it. And so all these pieces of DNA will be copied many times, but each time they'll end with one of these chain terminators. And we can separate these partial copies by size, and we can find what chain terminator was used to end each piece. And so we know what happens each time, how it ends, and we can reconstruct the sequence. This was very hard to do initially, but now it's automated. And so an easy technique that can be done in a standard lab with not too much trouble. Okay, we'll stop there and I'll go into more details on these next time.